So let's start our discussion with hallmark number 1, which deals with the aspect of immortality of cancer cells. And our discussion will be focused on the area of the chromosome that is called the telomere. So we've already covered about the senescence and the Hayflick's limit, but what we need to understand is that what exactly is a telomere? Telomeres are regions that are present at the end of chromosomes, and they are demarked by a specific sequence of repeated nucleotide bases, which are TTA, GGG. So if somebody says, how are we going to find out which region is the telomere? We find out by looking at the sequence of bases. So telomeric regions have this repeated sequence of TTA, GGG. Now the question is, why do telomeric regions shorten as the cell divides? And in order to understand that, I need you to understand the basic concept of DNA replication, which would require a basic concept of molecular biology. So I'm going to try and keep it extremely simple so that it doesn't get too complex for you. Every living being on this planet, whether it is human or animal or plants or bacteria or microorganisms, you name it, has the same 4 letters in their DNA, that is the ATGC, which are nucleotide bases. Now these letters are arranged in a very peculiar manner in such a way that A will always bind with T and G will always bind with C. Consider them like pieces of a puzzle. So these complementary pairs form 2 strands, and now these strands could be arranged linearly in straight line, or it could be in a circular manner. Human DNA is linear, that is coiled around itself, and the reason for coiling the DNA or wrapping the DNA is to protect it from any unwanted changes. Now whenever a portion of the DNA is copied, a little portion of the DNA unwinds and information from both strands is copied. However, there is a small catch. One of the strands is copied rapidly in such a manner that there are no breaks. Consider the phenomena that a DNA unwinds, information is copied, and the DNA winds up again. Now, the next portion of the DNA unwinds, information copies, and then the portion is wound up again. On the contrary, the other strand, which is a bit slower in copying the information, gets chopped off when the DNA closes, resulting in a small fragment of a copied DNA. So the final picture looks like a complete strand, while the other strand is in small bits and pieces with little gaps between them. So you've got one strand that is continuous, and you've got another strand that is in pieces. Now when you try to piece these strands together, you're going to find that the broken strand is smaller in length than a continuous strand, which means that some of the letters are missing in the broken sequence. But the problem is that you cannot add those letters because those letters require a hydroxyl group, and the missing information is in the area where there is no hydroxyl group, so you can't just add those ATGCs there. Luckily, we've got a special kind of Bob the Builder whose name is telomerase. Now, telomerase is an enzyme that is specific to the telomeric region on the chromosome. Now, telomerase has a sequence of letters that are complementary to the repeating number of bases on the telomere. So if the short repeating nucleotides were TTA, GGG, the portion present on telomerase would be AAT, CCC, or it could be AATC, or it could be TCCC. So when telomerase finds the matching sequence, it binds to the telomere and elongates it so that complementary DNA bases could be added on the other strand, protecting the shortening of the cell's telomeres. Now we understand that cells are avoiding Hayflick's limit by continuously repairing their telomeres, which makes them immortal. But what makes these immortal cells dangerous to the body? How do these cells contribute to cancer? Let's find out. Hallmark 2 of cancer talks about genome instability. And when we talk about genome, we are going to have to talk about the total number of chromosomes. Humans have 24 chromosomes, 22 autosomes, and 2 sex chromosomes. And every time a cell divides, these chromosomes are replicated and transferred to the daughter cell. This transfer of genome into daughter cells has to be error-free. Failure to do so could result in the genome being compromised, and there are a number of ways that could happen. Remember when we were talking about cell cycle, we talked about certain checkpoints that keep the genome integrity in check. The purpose of that is to make sure that the genome is copied as correctly as possible without any possible mutations. Now when I say the word mutation, it simply means that there is a change in the sequence of the letters ATGC. So it could be a change in one letter which results in a point mutation, or the mutation could result from the deletion of an entire section of the letters, or the mutation could result due to letters moving from one part of the chromosome to another. No matter the kind of mutation, this irregularity in the genome causes improper functioning, resulting in the development of a tumor. Another important terminology that you can see on the screen is called the loss of heterozygosity. Homozygosity and heterozygosity are basic concepts of genetics, and they simply mean that a gene has two alleles, one is dominant and one is recessive. If a gene contains either the recessive or the dominant alleles together, that gene is called homozygous. However, if a gene is a mixture of two different alleles, one dominant and one recessive, it is called heterozygous. The concept of heterozygosity is important because some of the diseases are homozygously dominant or homozygously recessive, which means that the gene cannot be activated unless and until it contains two identical alleles. In simpler words, if I have a healthy gene that is dominant and if I have a diseased gene that is recessive, the diseased gene will never be expressed unless and until the healthy dominant gene is lost. And since a healthy and a diseased gene are in combination, this combination is called a heterozygous combination. And once a heterozygous combination is lost, for this particular example that we're talking about, the recessive gene will come into play and our body will express disease. So now that you understand how the genome variation can lead to mutations which lead to disease expression, let's come back to the part where we were talking about the check during cell cycle and genome replication. It is important to keep in mind that mutations occur randomly and way too often, and that there are certain genes that keep a check on these mutations, known as tumor suppressor genes. Think of them as armed guards that are continuously monitoring genome replication. Anytime a mutation or error occurs, these guards respond to those errors either by terminating the cell or by initiating repairs. So when mutations occur in these tumor suppressor genes, these genes lose their ability to stop the cell from dividing during a mutation, and instead these genes become oncogenes. Think of them as good guys turning bad, and instead of killing the faulty cell, these oncogenes now enable those faulty cells to replicate and proliferate. Here is a beautiful depiction of what an oncogene actually feels like. Look at the horses pulling the cart. These oncogenes are responsible for the growth of cancer. So if you want to target cancer and if you want to control cancer, you gotta control these oncogenes. On the right side of the screen, you can see a list of oncogenes with the years that they were discovered. This slide was presented by Dr. Takmak, who is the founder of one of the therapies widely used to treat cancer patients. The next three hallmarks all result due to the loss of tumor suppressor genes and their conversion into oncogenes. When tumor suppressor genes transform into oncogenes, they are unable to identify growth suppressor signals, which means that they are unable to stop cells from dividing. One of the most commonly cited tumor suppressor gene is the p53 gene, which is also called the guardian of the genome. A mutation in p53 almost always results in a catastrophic outcome. Another tumor suppressor gene is the retinoblastoma gene, abbreviated as RB, and we are going to focus on this gene in hopes to understand the hallmark number 3. Both of these tumor suppressor genes checks the cell in G1 phase and stops it from getting to S phase if there is a possible mutation or an error in the genome. In the figure, you can see that the RB gene is bound to the E2F factor. As long as RB remains bound to E2F, the cell cannot enter S phase, and thereby replication is prohibited. However, on the occurrence of a mutation, the RB gene is no longer bound to E2F, which is a transcription factor. In the case of p53, if it finds that the cell's DNA is damaged beyond repair, the p53 gene induces apoptosis, eventually killing off the cell. By killing the damaged cell, the p53 gene stops the cell from spreading the abnormal damaged DNA to other cells. However, if the gene is mutated, the cell does not undergo apoptosis and hence continues to spread damaged DNA. Now, p53 is no longer inducing apoptosis and abnormal cells aren't dying. This mechanism makes cancer cells resistant to death, and as the number of mutations increase, the resistance to cellular death also increases. And this occurs due to the activation of pro-survival genes like the BCL family and the inhibition of pro-apoptotic genes like the BAC and BACs. So up until now, we have mutations in the genome, we have loss of checks, and we have loss of apoptosis. And since there is no check and balance of the cell continues to grow. So this unobserved, uncontrolled growth pattern of cells lead to hallmark number 5, which is sustained proliferation. Now the cells are receiving signals to grow because no cells are receiving signals to die. And there are no checkpoints because those tumor suppressor genes are now oncogenes and they are promoting growth of cancer cells. But there are also growth factors that the healthy cells are receiving, and in response to oncogenic cells, these cells are also growing. So what we're left with right now is a massive chaos of uncontrolled cells in the human body. And in our next cancer hallmark video, we are going to have a look at what do these uncontrolled cells do to become cancer.